Computer. Okay. So, hi everyone. We're gonna start now. Ben's gonna start. I'm gonna grab a laptop.
Everybody's rolling in. So guys, how are we feeling about the mini midterm? So terrified?
Yes, no, maybe so.
So terrible.
Room
Excellent.
temperature, not so
Wherever
good.
you're at, we'll figure it out. So my recommendation as we go through these slides, because this is basically just a review of the material that you should know, is to just take notes on what you need to know from the material and then use that to further study, kind of figure out where you're at. A lot of the material, if you've taken her quizzes, which of course you have, it's going to be the same approach. So it's all open response, it's all, you know, your basics, and then you're going to be answering more complicated questions, knowing the basics. So it's going to be very similar. The reason she does the mini midterm is to prepare you for the midterms, to kind of give you an idea what that looks like. You know, you're writing a lot, you're doing a lot of that kind of stuff. So getting comfortable with that. I personally, I've taken some of the really long exams, and I'd recommend getting something that you can write with for a while because hands do cramp. So that's a recommendation, but you probably won't need it for this one. So those are kind of my thoughts, kind of preamble before we go into this. So here we are. Perfect. So prokaryotes and eukaryotes, how do we feel about it? So we've got the prokaryotes and eukaryotes. The way I like to think about them is versus each other. So you've got the, you know, single organisms, no membrane organelles, has a nucleoid, but not a nucleus. And it's just to thinking about these comparisons. That's the important part of looking at eukaryotes versus prokaryotes. And she probably won't have it in this material. Viruses, again, small genetic material inside. I always think of, hey, you know, this is the little thing with taxi, and it keeps it in your head. You're up.
Oh, okay. So this is transcription. We talked about this a little bit today, not too much. You're not going to need to know as much detail though as we're going over in class, because Unit 3 is more on transcription, translation, and everything. You just need to like know the general basis of it for tomorrow's mini midterm. So you just need to know that DNA turns into RNA, which is facilitated by RNA polymerase, right? And does someone want to tell me how that happens? Kind of quick general overview of how that happens. Sure.
It goes into the DNA is RNA polymerase attaches to the DNA double helix and is unwound, and RNA bases interact with the DNA and build as a template and out as a string of RNA.
Yeah, that's basically it. And they have the opposite base pairing, right? And you all know that, and it gets built, and we all talked about that, right? Of course. So in eukaryotes, this only takes place in the nucleus, right? Because eukaryotes keep their DNA in the nucleus, right? And in prokaryotes, it takes place in the cytoplasm. Why? Someone, just shout it out. Yes, no membrane-bound organelles. They're all just floating around in the cytoplasm, which is what I said there. Everything is in the cytoplasm. Here's a little diagram to show you about transcription, which is basically what we've already established. So the RNA gets made because it gets unwound from its double helix form, right? So they're separate now. Here's your template strand, and you add bases to make mRNA, right? Yep, everyone good? Does anyone have questions about this? Is anyone confused about the general idea of transcription? Good. Okay, translation, the second part. mRNA, right, that we just made from transcription, This is the process of turning that mRNA into an amino acid sequence. So amino acids are the building blocks of proteins. Is everyone good with that concept? Right? So amino acids are smaller and you put them together and they make proteins. After amino acid sequence is formed, it will fold into a three-dimensional protein. Right, because all of these molecules are happening in three-dimensional space. You have to remember that these proteins are going to be three-dimensional and the amino acids are going to create like either like a barrel, like a beta barrel, or an alpha helix, or like a globular protein. It's three-dimensional. You have to consider the shape of these molecules because the structure of their protein determines the function of the protein, right? So does someone want to tell me what a beta barrel might do? Any example? Yes. Yeah, it might sit in the cell membrane and allow things to move through it. Good. Does anyone want to tell me an idea of like what a globular protein might do? Okay,
It forms this cell wall. Or-
not quite. Cell walls are made of, does anyone know what cell walls are made of? Yeah, phospholipids. So cell walls are not quite proteins. They're phospholipid bilayer. But so globular proteins might do anything because, and that's kind of the answer I was looking for. Like they might do anything in the cell depending on their specific structure, because they're just going to be different shapes and they'll really just do anything, and you have to look at the shape of the protein. It depends. That's basically the answer to that question, is it depends. So anyways, do I have anything else on this slide? Oh yeah. The ribosome does translation, right? Yes, the ribosome is technically two globular proteins, right? So that's an example. It does translation and it takes place in the cytosol, always, right? Does anyone want to explain why? Someone new? Okay then. So it takes place in the cytosol because ribosomes are located in the cytosol, right? They might be on the outside of the endoplasmic reticulum, as we talked about in class, but they're never going to be inside of the endoplasmic reticulum. Sometimes the protein moves into it as it's being translated, but the ribosome is always outside of the endoplasmic reticulum in the cytosol, and proteins are always translated in the cytosol. Yes? Cool. Any questions about that? Okay, solid. And this is just a diagram of what happens during translation. Translation. Yeah. So you've got your tRNAs, which base pair, and they have their amino acids, and it makes a protein, right? So the ribosome turns your mRNA sequence into a protein, which then folds, right? Yes, good. The tRNA anticodones in the ribosome or in the cytosol. No, the tRNA is always going to be bound to its own anticodon, but which will then be attached to a new protein or a new amino acid, right? And you'll talk about that a lot later. You'll get to that. You don't need to know that quite yet. Yes. But yeah, basically, tRNA always is going to have its anticodon, not always going to be attached to the amino acid, but it will always get attached later to the same amino acid. Yeah, but you don't need to know that. Good question. Ben's turn.
So just to remember simply that gene expression refers to the combined steps of transcription and translation. Big picture. So differential expression. So nearly every cell in your body contains an identical copy of your entire gene. So basically, when you get a cell, this whole process it goes through. So at the beginning, it can be anything. You're just reading from the code book. And as you go through these stages, different components are taken out or chosen, and then it breaks it down. And once you get through that phase, it becomes a specific cell, so a liver cell or a skin cell. So difference, like I said, is when and which genes are expressed. So this is where Trying to remember, have you guys gone over, introns, extrons? Okay,
Introns and exons.
excellent.
That's, topic,
Okay,
Yeah.
that's the word.
yeah, that's.
So you don't need to know that yet,
Yeah.
but you all should know that now.
So basically, when. I'm gonna put it pretty simply since you don't want to stuff too much in your head, but basically parts get cut out that aren't gonna be used. And that's how you get this level of differentiation.
So basically, there's always the same sequence, but it's not able to be used based on like its ability to have some machinery that cuts it up specifically and fixes it to the right form.
Correct, so when the DNA, when the DNA becomes RNA, that RNA is then taken and cut up by the spliceosome. And then that's, and even within that, only certain portions of that are actually expressed. So it's kind of like a cutting down, you can actually get multiple expressions of certain amino acid sequences. And that's why, like for instance, I'll get to that in a second, but for instance, like a liver cell could be two of one set of amino codons and then two of a different set. And it's how things are combined anyways. So are we comfortable with that? Okay. Yes.
So do you want to put the genes that are in the cell that aren't used, they just stay there?
So they get cut up and degraded. They get
The
degraded.
genes in the cell, all of your cells have the entire genome,
Mhm.
right?
Right.
All of the genes that all of the chromosomes that you have are in every single cell's nucleus. If you go back a slide, one exception, there are exceptions. One of the exceptions is like a red blood cell, and they eject their nucleuses. So they don't have any of your genome in them.
So the question is, are you talking about DNA or RNA?
DNA.
DNA, so yes, that does not get cut up.
Okay.
That's just used to and get your RNA. The RNA is what gets cut up,
Okay.
and then it gets exported into the cytosol, where it gets turned into a protein.
Thank you.
Yeah.
So RNA is the process of differentiation.
Kind of. An example might be like if one specific DNA sequence codes for a protein in a liver cell, the same DNA sequence might code for something totally different in like a skin cell, right? And the difference happens when you do intron splicing, because that whole sequence will get translated by the RNA, will get translated by the RNA polymerase, right? And it'll leave the nucleus, or it doesn't leave the nucleus first. It gets spliced first. It'll get spliced first, and then it'll leave the nucleus. And that sequence that leaves the nucleus will be different in a skin cell or a liver cell. And we'll actually talk a lot more about this later. You don't need to know the details of this yet.
So you're talking about RNA processing in eukaryotes.
Yes.
Yes. So. Different molecules, the way I like to think about these is, I think of the word poly, which is the word many, so it allows you to just look at the words, okay, poly, so you know this is gonna be a lot of whatever it is. So you've got, a polymer, long molecules made of repeating units of amino acids. Then you have your examples, DNA, RNA, proteins, carbohydrates, a peptide, again, a short chain of amino acids. You got polypeptide, so again, putting the peptide and the poly together, all you need to know is it's just longer, the word poly, that helps me to remember that. And then proteins, again, are a string of amino acids. So, and again, the distances are different, but the average is around 200 to 400 amino acids.
I would say you should be familiar with these numbers, right? Like you should know that a peptide is really small. You should know that a polypeptide is anything longer than a peptide, and a protein, and like the general idea, but you don't need to like know the numbers. You just need to conceptually understand the relative lengths. Yes? Got it? Yes, no, maybe so? Cool.
So monomer, peptide, polypeptide, protein,
Yes.
and then
Yes.
polymer, just like the umbrella term for
Basics.
like.
I actually have a slide about this. This is a cool diagram with my horrible handwriting that I drew earlier today. So, atoms make up molecules, which are kind of the same as monomers, right? Because molecules are groups of atoms, monomers are a unit, which is made up of groups of atoms, right? A one unit. And polymers are made up of many molecules or monomers, right? Yes, is everyone good with the top half of this chart? Any questions? No. So then here's our specific example that is actually very relevant to you in this class. Atoms that you might see in this class, carbon, nitrogen, hydrogen, oxygen, etc. You'll see more than that, but these are like the main atoms they'll see in this class, make up amino acids, which are a type of monomer. And amino acids make up proteins. Proteins can be either monomeric or polymeric, which means they can be like one unit or multiple units. Right? Yes. Does anyone have questions about this?
We talked about proteins being polypeptides. The number of, like repeating nucleotide sequences, how can it be monomeric? I think just one polypeptide,
It's kind of related to quaternary structure. Can someone tell me what quaternary structure is?
polypeptide chains building together.
Yeah, it's multiple polypeptide chains. It's talking about a protein with multiple subunits, right? So that's why it can be either monomeric or polymeric, because it could be one really large molecule, which is referring to a monomer protein, or a polymer protein, or polymeric protein, which is multiple subunits. It's kind of confusing.
So monomeric would be tertiary, whereas polymeric would be quaternary.
Not necessarily. A monomeric protein you would say that it doesn't have quaternary structure, but a polymeric protein, you would say it does have quaternary structure, because not all proteins have quaternary structure. You look a little confused.
So the definition of tertiary structure is a single polypeptide chain, defined by the R group interactions, and like that's how it's structure equals function, or yeah, structure equals function.
I like to think of it as secondary structure is formed by the backbone interactions, whereas tertiary structure is formed by the R group interactions. That's the difference, and both of them build towards the shape of the protein. Yeah, both of them determine the shape of the protein. We're actually going to talk about amino acids a little bit later more. You can save your questions for that, because I think we should keep moving. That's very relevant. I think it might make a little bit more sense later. And here's a slide about important organic molecules. So amino acids are an amino group, a carboxyl group, an alpha carbon. I'm standing in your way. An amino group, a carboxyl group, an alpha carbon, right? And an R group, off of the central carbon, the alpha carbon. It'll be referred to either way, alpha or central, same, it's the middle carbon. Okay, nucleotides, nucleotide, pentose sugar, this one in the middle, this is your pentose sugar, because pentose kind of has the prefix of what? Like a pentagon, yeah. So, and it looks like a pentagon, so that's how you identify your pentose sugar. It has your phosphate group, which hopefully everyone recognizes because you're gonna need to know that, PO4 phosphate group, and your nitrogenous base. And nitrogenous base comes from the fact that it has a lot of nitrogens in it. You see these nitrogens, nitrogenous. Yeah. Those are the building blocks of RNA or DNA. Phospholipids are, they have your hydrophilic head, which likes to bond with water, and your hydrophobic tail, which is a lot of non-polar bonds, and therefore it does not like water. And that's what makes up the cell membrane. And we talked about that already. And then carbohydrates, 5 or 6 rings usually, right? 5 or 6 sided rings with 1 oxygen atom. So most of them have carbons at its points, which is, we talked about this today. There are always carbons at the points in OChem shorthand. And 1 oxygen atom, which is always bronze, and that's the source of energy. And that's what a carbohydrate looks like. I don't think these are too important, but yes.
I thought that amino acids also had a hydrogen group. Is that true too?
A hydrogen group.
Yeah.
I would
That's
call
what
it a
I
hydrogen
know, but I
group,
just wanna confirm.
but I would call it a hydrogen bonded to the central carbon.
Okay.
Yeah, it's not like a group of its own, because only one of the amino acids, glycine, has a hydrogen as its R group,
Mhm.
which might be a case in which it's appropriate to describe that as a group,
Mhm.
but it's not really, I wouldn't call it a group, it's always just a CH,
Okay.
and it's bonded to the central carbon.
Gotcha,
yeah.
thank you.
So quick question. DNA or RNA? All
Good question. DNA or RNA?
Excellent.
Y'all so smart.
Which one's this saturated, unsaturated fatty acid? No, the pink one or the green one?
Pink.
Pink is saturated?
Oh, saturated or unsaturated.
Aren't
So
they?
saturated is the, if I remember correctly, it's this one, and unsaturated is that one, because double bonds prevent them from being able to bend, and so they can't.
So unsaturated is straight, pink is
No.
saturated.
This is saturated, this is unsaturated.
Would we be able to know what amino acid is by looking at the carbon group or
The carbon group?
sorry?
That, you're saying just the basic amino acid?
Yeah, like would we be able to tell each of them just based on?
Are you, oh, you're asking if you should know all of the amino acids and be able to identify them based on the R groups? Not necessarily. You don't need to know that, especially not in this class. You might need to know that later if you're a biochem major or something. You don't need that for this class. But what you should be able to determine is where the amino group is and where the carboxyl group is.
And do we also need to know each nucleotide and be able to identify it based on its chemical structure?
No. No. And you also probably won't need to know that much about the phospholipids either. Like, I don't know which one is the saturated and unsaturated tail, and you'll never need that information probably. For this class, you'll never need that information for this class. You might need it for another point. That's not, don't hold me to that. Okay. Is everyone good on that? Because now we're moving on to topic 2. Yes. Differential gene expression. Yes. Transcription factors, right? So that's actually another, that is actually another thing that will determine your differential gene expression. Transcription factors sometimes will regulate, upregulate or downregulate the rate at which a gene is being expressed or transcribed, right? Which is part of the when in which genes are being expressed, right? But you don't really need to know that for tomorrow. That's like a topic 3 thing. So we can talk about that later. Kind of. I would just be familiar with the term, with the idea.
We're not gonna have to actually.
Yeah, you're not. You just have to be familiar with the idea that the cells are not expressing their genes all at once.
Yeah.
That's a really important concept, right? Cells don't express their genes all at once. They don't express all of their genes, right? Cells are only expressing certain genes because they're supposed to do certain functions. Yes.
And limitations with the spatial limitations, with the cell needs and a machinery use. So obviously, if you're trying to express 2 parts at the same time, you use the same machinery, so you can't do that. And then limit like what the cell needs, and then spatial is just the.
Yeah. Yes.
Those are the only limitations.
I think. Sounds right to me. Okay. So topic 2, amino acids.
All right.
All yours.
I love amino acids.
I do
So
too.
I think the really important thing to know about amino acids, kind of following up on what we were talking about before, is charge, polar, and what kind of charge they have. And it's really based on whether there's a nitrogen or an oxygen and the relationship between the atoms that are bound to the nitrogen and the oxygen. So this is the basic chart. She will always give you either this if you need it, or the specific amino acid on there. So you don't need to memorize this. But again, what I said before, the kind of the easy way is to use more chemistry knowledge and understanding, oxygen bound to hydrogen. What's the difference in electronegativity between those 2 atoms? You're good with that?
That's the question about. As far as with the charges, are we gonna have to know how we make the charge that we have out maybe in the percent? Or is it better to have the decimal side? Like, let's do the charge.
It should, if she puts this on here, it will look actually just like that.
Just like that.
Yeah.
You will
So
get
this-
exactly this chart.
Okay.
Or she might give you that spectrum chart, which is like the most hydrophilic, hydrophobic ones, or the in the middle, like moderately hydrophilic, hydrophobic, right? She might give you that. She might give you both. It depends on what questions. You'll be given all of the information you need to answer the questions without having to do an excessive amount of memorization.
She's not big on memorization.
Yes, that's correct.
So, and also when representing, this is something we'll learn Gen Chem organic. Whenever we're representing a molecule at all, you have to put the formal charges on. So it would be incorrect for them to
Represent
show them
without the
any,
charges.
yes, exactly. And that whole, the gradation between the amino acids of polar to non-polar, you can basically guess which ones are, polar in charge because they're going to be at the very bottom. Alright, who we got? Okay, so polypeptides, how they're bonded together. let's see. Well, I guess the diagram basically shows what yeah. So the hydroxyl group or the carboxyl group and the amine group come together. They have a dehydration reaction. You can think about dehydration reaction as you're getting water out of it. So the water's coming out of it. Honestly, the way I think of it is a guy running through the desert and all the water's evaporating out of him, and then it creates a bond
That's
between these two.
I like that.
And so the way well, I'll show you that on the next slide. Sorry. But the way you're able to tell is it's always the double bonded carbon to oxygen to the nitrogen. And the bond between the two amino acid is called a peptide bond. It's a covalent bond, which is very important. And it is catalyzed by a ribosome. So any questions on that?
You should all be familiar with what a carboxyl group looks like and an amino group looks like. It's not super like memorize-y. That's more just like having looked at it enough times to know what it looks like. You know what I'm saying? Yeah. You really want to practice that. It's really confusing.
So, question. There's that bond right there. So you're always looking what I said before, kind of reiteration. So, and then just take a minute, see if you can find the bond in
Yeah.
this example.
So I want you to think about how many peptide bonds are in this structure and also how many central carbons there are. And I want you to be able to point them out to me. And I'll ask some people to come up and point them out.
We have a volunteer for the peptide bond.
Someone from over here who I've not heard from.
Yeah.
All right.
This one,
Right. And how do you know?
because that's like where the hydrogen, like this is, how do I explain it? Like this is where the hydrogen was lost, and this is where oxygen was lost.
Nice,
And then
Yeah.
nice, good job.
All right. Everyone hear that? Yes? Everyone on that side of the room? You didn't hear it. That's what I thought. So she just said I'll come over here. Space over here. So she just said the peptide bonds are here and here. And how do we know that? Does anyone know? Yep.
There's one carboxyl group and the.
Yeah. Yes, we're wondering. Yes. So the carboxyl group had a double bonded C, carbon and oxygen, right? And the amine group had a nitrogen and a hydrogen, and there was a hydrogen loss to this, bond formation, and then there was an OH loss, which formed water. 2 hydrogens and 1 oxygen, water. Yes? No, maybe so? Good.
So somebody from this side of the room, central carbon.
Yeah.
Mhm.
Okay.
And,
Mhm.
Okay.
yeah, it's that one.
Yep.
How do you know?
It's kind of in the center of everything, it's the raw oxygen groups right there. That's the amino group, that's the peptide bond right arm, so it's kind of, and that's the other thing. So kind of-
Yeah. Yeah, you find the R groups. Good.
Good trick.
Right, you find the R groups. And they're bonded to the central carbon. Yes, anyone have questions? Anyone have concerns? Anyone have
Errors?
comments? Good,
Excellent. All right. Peptide structure. So N terminus, C terminus. I know it's on here, but who remembers what they are?
The NH3 is the N terminus, and which can also just be NH2. And then the C terminus is C double bonded O. And OH, but you lose that H and have a negative because it has an equilibrium within an aqueous solution, or no, because of the acidity of the cytosol. So the N terminus will add an H, getting a positive charge, and the C terminus will lose an H, getting a negative charge, but they still remain the same as their uncharged form, so it doesn't affect any backbone or any other thing. Yeah.
So another question, when we're counting, where do we start? Do we start in the N terminus or the C terminus? Excellent. N terminus.
Yep.
I think you explained the slide to me, so, you know. Good job. So our groups are the part, of course, that's different between each of these, and kind of going back to the amino acids. So, you know, our group, and the R group is what differentiates between the different amino acids. Really, the backbone's exactly the same. Of course, besides the ends, because you get the different groups singly on the ends. All right, basic chemistry. Yeah, I love chemistry. Yeah. So covalent bonds, ionic bonds, electronegativity. Very important. Once you know them, it'll all of your amino acids, just looking at them, it'll help you to understand, hey, this one will do this one will do that. Super important. So covalent bonds, electrons are shared between the two. It creates a stable bond. The reason for that is other molecules like water can't come in and steal the electrons away from that bond. And so they're really good for structural purposes. That's why the backbone, the polypeptide chain is made up of covalent bonds. That's why they're important. Ionic bonds, now, this is when you've got, like for instance, with salt, you know, you throw salt in water, right? It dissolves, but it's hard when you've got table salt. So basically, when you put it in water, the water molecules come in and steal that electron off. And so that's because the ionic bond, that electron is actually being stolen by whichever really needs it to complete its octet, which I don't think you guys have done it.
Who is in general chemistry right now?
Yeah.
Raise your hands. I want to see how many, like what percent maybe? Who's in general chemistry?
Like
Okay,
some people.
not a whole lot, but a decent amount. If you're in general chemistry, I would recommend not thinking about general chemistry too much. You want to think about cellular chemistry a lot more than what you're learning about in general chemistry. One example is that in Gen Chem, you're taught that ionic bonds are stronger than covalent bonds because of the full transfer of electrons to form a complete octet. That's not entirely relevant. A lot of people have come up to me and asked, should I talk about the formation of a full octet? And honestly, I didn't think about that once when I was taking this class. You don't have to mention it. No one is looking for you to talk about the octets or chemistry and also ionic bonds. Not stronger in a cellular context because of water, which is made up of oxygen, hydrogen, and the nature of that bond. And that's kind of it.
But it is important to know because
Yeah.
you're in a aqueous environment. Your
Yeah.
body's full of water. So a lot of this stuff is very logical. It's very, oh, hey, if I think about the bigger picture, so if you get stuck on a question, ask yourself, hey, in the human body, what would this look like? And oftentimes that can get you the right answer. And then you just have to explain it that way. Okay, so electronegativity is we really only deal with sulfur, nitrogen, oxygen, carbon.
For what?
I was just thinking of the
For electronegativity.
yeah.
I think the atoms you need to be most concerned about are nitrogen and oxygen. Those are really electronegative, right? And then carbon, hydrogen. Those are also important. Honestly, there's not a lot of other elements that you're really going to see that often in this class. Sometimes you'll see sulfur. Sometimes.
Yeah.
And that's about it.
Once in a while. it does.
Electronegativity with sulfur.
I don't think you really need to know the electronegativity of sulfur. I'm not gonna lie. Like the number specifically.
So basically, the farther you get
Yeah.
along this chart, it's higher.
Yeah.
Towards the corner. So I think of sulfur. Sulfur's really big. So even though it has a pretty decent electronegativity, it doesn't pull a lot. So it doesn't have the same effect as oxygen. Really, what you look at here is oxygen and nitrogen, because oxygen basically just will make a ton of polar bonds. So you're always looking for the oxygen in the molecules that you're looking at. Any questions on this before we move on? Great. Okay. Covalent bonds. Well, I did go over some of this already.
Oh, yes. I already gave my spiel about caring more about covalent bonds than ionic bonds.
Let's see. Yep, we talked about that already. Don't get too big into it.
Someone describe covalent bonding and the difference between polar covalent and non-polar covalent. Someone from your table. The difference between polar covalent and non-polar covalent.
Sort of different electronegativity between the two it's atom. So the non-polar covalent bonds usually have two that have like roughly the same electronegativity. So this will be the carbon-carbon, they're the same, so they would sort of share their electrons equally. But then for the polar covalent bond, one of them has a stronger electronegativity than the other one. So the sort of electron they're sharing spends more time on one side and creates like a partial positive and partial negative charge.
Yes. Yes.
So to simplify that, the way I like to look at it is if you have the same molecule, or if you have something that has similar electronegativity, you've got having a tug of war, you've got somebody on each side and they're pulling at the same rate. Now, if one is much stronger than the other one, you're gonna get a dipole, and one side is gonna be much stronger than the other one. So that's a great way to just keep it in your head.
Okay, so when you're talking about polar covalent and non-polar covalent, you're thinking about hydrophilic versus hydrophobic, and this is in context of a phospholipid. So does anyone want to come up and point out the hydrophobic area on this phospholipid? Someone from this half of the room that I have not heard from yet. The back table, perhaps.
Hydrophobic,
The hydrophobic of this one.
oh, that,
Yeah,
that,
yeah, the zigzag area is the hydrophobic part. Do you know why?
Because those are like carbon chains, that are not photo.
Yeah. The hydrocarbons, yes, correct. So I given you this diagram because this really breaks down where the hydrocarbons are and where your electronegative atoms such as oxygen are, right? And this is basically the organic chemistry shorthand form of this. So all of these are hydrocarbons, carbon bonded to hydrogen. Is everyone cool with the term hydrocarbon? Yes, good, awesome. So because of that, these are gonna be really, really, really non-polar, right? Because CH is a non-polar bond. They're gonna share the electrons pretty evenly. And so basically what you get is like this whole area that does not want to interact with water because water is charged, right? And if water is charged and this is not charged, it's not gonna have that opportunity to like interact. It's like if I had a magnet and I put it on a magnetic surface, right, and it sticks, that's interacting with the magnetic surface. But then if I held like a marker, I don't have a marker. I was thinking this was a whiteboard. I'm not gonna lie. But then if I held up this piece of chalk next to this whiteboard, right, what's it gonna do? Right, because it's not magnetic. There's no charge in this piece of chalk, which means it's not gonna interact with the whiteboard. It's kind of like that. It's a charge thing, but with molecules. I mean, those are molecules, but with more organic molecules. Okay. Is everyone cool with the phospholipid bilayer? Yes?
Yes, so wait, like this whole part like up here too is even hydrophobic?
Yes.
Okay.
So actually, someone from over here now. Do you want to point out the hydrophilic part? Yeah,
Kind of like that's it. That's, right?
basically.
Right there.
Yeah, it's the part with oxygens.
Yeah.
All the electrons.
Oxygens.
Yeah. You want to look for oxygens and nitrogens in this one.
Is it just the piece surrounded by oxygen that is attached? Or which part, where's the tail end?
Right, yeah. So in this one, yeah, the head is kind of this area. It's mostly centralized around here because that's where this phosphate area is, right? But this is still gonna be a little bit charged, but this is mostly where it's gonna be, especially because you see this negative here, right? But then and the positive there also makes it want to interact with things. Either sign actually will make it want to. It's just when you have no signs, no chargers. And so this whole area here, mostly with like these squiggly tails, that's the tail area. Right.
So in the top picture, the CH there, CH. Are those specifically like tails?
Yes, yeah. Everything below here is the tail, everything above is the head. Yeah, yeah. Any more questions?
Yes,
So those oxygens, like, it looks like it's not. Then it should start, but those are still starting.
but these ones here,
Yeah, those oxygens.
yes,
Okay,
Yeah.
Yeah.
whole chunk, not just the phosphate group that will check.
yes, correct.
Yeah. Okay, any more questions? All right. That's it, yeah. And then this is the second version that I have here about hydrophilic versus hydrophobic. And now we're gonna think about the same concept, but with amino acids. So the nonpolar amino acids are, everyone say it. That was terrible. It makes me so sad. Okay, and the polar version, the polar areas, the polar amino acids and the charged amino acids are hydrophilic. They love water. It's like when I put a magnet on a board, on like a whiteboard, right? It's gonna stick to the whiteboard. So that's basically what's happening. It wants to form, it's gonna form interactions with the whiteboard and it's gonna do things. But or like when you put two magnets together and they repel each other, that also is an interaction, right? Yeah, it does things. You have a question?
So the van der Waals are the weakest type of bond, then why aren't they hydrophobic?
Van der Waals, I would not consider as necessarily a bond. Van der Waals are really, really weak interactions. You were saying that. Do you want to go ahead? Do you have something to say?
Van der Waal interactions are also, LDF, which is
Yes.
London dispersion forces.
Yes.
It talks about,
Wait, I want to correct that. So there's a bit of confusion about van der Waals. Van der Waals actually mean electronegative forces in general. It's an umbrella term. But London dispersion forces are what people get the two terms confused. London dispersion forces actually mean
but London dispersion forces are the weakest type.
correct.
Yes.
Yeah, you're correct.
London dispersion forces are temporary dipoles created by, fluctuation in electron movement.
Yes.
So our electron clouds are constantly like moving, or the electrons in the cloud are constantly moving.
Yes.
And so most often what will happen is there'll be a cluster of electrons and that'll make a temporary negative charge. And on the other side, it would be a temporary positive charge.
Yes.
These temporary dipoles create a temporary interaction. Just, I would recognize that because it's temporary, it's very weak,
Yes.
rather than a stable,
Yes.
dipole that's created by
For sure.
photons.
That was a really good explanation. Did everyone hear that? Did anyone not really hear what she said? Awesome, because that was really great. Thank you. Basically doing my job for me. I don't even have to do anything. I just have to stand here and look good. Okay, so you can recognize the nonpolar R groups because they're mostly CH bonds, right? Because we said that they only have that hydrocarbons are really nonpolar. So all of these mostly just have CH groups, right? And you can recognize the polar ones because they're going to have charged molecules like nitrogen, right? In your positive charge nitrogen or oxygen, or both nitrogen and oxygen, right? And so that's how you can recognize them. If you don't really see any of them, any of those charged molecules, it's probably nonpolar. And in a lot of cases, you're going to be given an amino acid and told what its charge is, or not what its charge, like what its characteristic is, right? And then you'll know, but in some cases, you might have to know. You know what I'm saying? So, yes.
So for non-polar versus polar, non-polar is non-charged, whereas polar is
Yes.
charged.
Yes.
We see the separation between positive and negative.
Wait,
Well, polar is not necessarily charged.
yeah. polar
Only the
isn't
charged ones.
Only, so
are
charge
charged.
means that it has that formal
Yeah.
plus or minus on
Yeah.
one of the
Yeah.
atoms. So just so you
Yeah.
don't say the wrong thing. Polar
Polar
is
is just the 0.4
Yeah,
above 0.4.
exactly,
Yes.
but up
Yes.
to, what is it, to 1.1. It's like, I think it's like up to one, like
Yeah.
one distance, and then after one it becomes charge.
Oh,
So,
Yeah.
and you can look that range.
Yeah. But yeah, the charged ones have polarity to them, but they're not called polar, they're charged, but the polar ones are not charged. Yeah.
Okay.
So these are basically a type of polar amino acid. They're just more specific and stronger. They have stronger charges. Yeah.
Yes.
Better interaction. Yeah. Is everyone good on this slide? Yes.
So, for the van der Waals, it can only happen between polar bonds if they're not charged, or I meant if they're charged, right? So van der Waals can happen between uncharged polar bonds.
Yes.
Okay.
Yes. Yeah. So is everyone good on the concept that electrons are always moving? Right? Everyone knows that? Yeah. You were gonna say something.
Well, I was gonna say that you actually can get van der Waals technically with non-polar.
Yeah.
Because basically, think about it. You got the earth and the moon's moving around, it's pulling the oceans, right? And you get exposed shore, and then that exposed shore would be. But basically, you're getting this group body of water is getting pulled, and then you can say, well, on the other side of the earth or on the other side of it, you have more exposed
Right.
beach, and then that side will attract something. So it's about, you've got this one molecule here, something over here causes all of the electrons to spend a little more time on here, on this side of it, which makes this side more positive and this side more negative, not to do with what's over here. And then another molecule will be like, ooh, and then it's you're gonna get some negative on this side to match the positive on the other, and that's, you get this big chain. They're very ineffective, you know, until you get them in large groups, and that's generally where it's,
Okay.
is in large groups. So van der Waals, this is our slide factory. But I guess, no, that is an umbrella term, just kind of making it an example, but it should say London.
Okay.
Yes.
Yeah, well, that's good. You have a question?
London.
London dispersion,
Yes,
yeah.
yes.
So London dispersion forces have polar and non-polar.
Yes,
Okay.
London dispersion forces occur between all molecules.
Yeah.
Everything that has electrons has the ability to have London dispersion forces. Yes? Yes. Yep. Oh, no, I got it. That's it. Is this the last slide? Yeah, this is the last slide. When it has more oxygens, it is the hydrophilic head. Yeah, you can identify the hydrophilic head by the presence of oxygens.
Yes.
Because you know that oxygens are electronegative and they want to interact with water. Yes.
Correct.
Yes. The presence of the CH is what makes it hydrophobic.
General description of van der Waals is what again? I'm sorry.
General description.
It's just an umbrella term. For like polar, charge, all of basically, anything within this polar category, so polar charge, positive charge negative. Dipole, ionic,
How would you define it?
how would I define it? Terrible, yeah. Let me,
You should have your own laptop.
Yeah.
Most of my devices.
I'll
are
look that up and we'll get to it.
You can like look it up honestly yourself. If you have to look it up, you can just look it up yourself. Honestly, van der Waals, knowing that London dispersion versus and van der Waals are very similar is, I think,
She mentioned this in her class.
really, what did she say? What did you want them to say?
She said that she had been under the misunderstanding. that
I remember
Van-
this.
Yes.
Yes, that they were the same thing. They're
And
not
they're
the
not.
same, but they're similar.
No, this is just the Van der Waals, it's just. Umbrella term.
They're just about.
It's
They're
just. an
just
umbrella
charge interactions.
At London dispersion, what we've been calling London dispersion.
Yeah.
groups, they're just London
Yeah,
dispersion.
I think it's an umbrella term for intermolecular forces.
yeah,
Yes.
I
It
think that's,
is.
maybe that's what she was confused with.
yeah.
Okay.
Yeah.
The
it talks
London-
about
Yeah.
Electrostatic forces that occur when atoms or molecules are very close together.
Yes.
So charge.
Yes.
Yes.
Okay, we're gonna move on because we don't have that much more time left. But if you have questions, please talk to us afterwards. Okay, amino acid polarity, that's me. Okay, so you've all seen this scale, right? Yes, you're all familiar kind of with the hydrophilic, hydrophobic spectrum of amino acids, right? Because it's not like a black and white. Some amino acids are more hydrophilic and some are less, like we were talking about earlier. In the polar ones, the polar amino acids are mildly hydrophilic usually, whereas the charged ones are like really, really hydrophilic. And it also is context dependent. Does anyone remember the things that might determine whether or not, or what things might change how hydrophilic or hydrophobic an amino acid might be? You- What?
Heat.
Yes, heat. What else? pH? Yeah.
Light,
I was just saying salt.
is it light?
Light?
Yes.
Yeah. Okay. Yeah,
Yes. Nice.
yeah. So there's a lot of things that will change the context of this, right? And you should be familiar with them. Some questions that you should ask yourself are, where are the most electronegative atoms? What other amino acids are near the amino acid that you're looking at? What type of secondary structure is a part of, and what is the function of the protein overall, right? And there's a lot of questions you could ask yourself. You just have to read all of the words and be really careful with what the question is asking and what it's telling you. I would recommend annotating your questions, right? If they ask you something about amino acids, because all of the words are important, kind of, in their own special way. And you need to know what is important to the context you're talking about, right? Okay, so I have two amino acids here that are kind of in the middle of the scale. We have tryptophan and tyrosine. Tyrosine. Is that threonine? I think this is threonine, actually. I might be wrong though. I might have just lied to you. Don't trust me on that. But this is definitely tryptophan. This one has a nitrogen in the middle of this ring structure, which makes it less non-polar than other ones. Because the nitrogen is very electronegative and will draw electrons toward it, but only sometimes due to the large ring structure of this molecule. So it depends, but that's why it's in the middle. This one is generally categorized as polar, but because of this larger ring structure, it might also kind of be non-polar sometimes. Because a lot of these are all hydrocarbons, and these will be really non-polar, but then you've got this OH at the end, which makes it kind of polar. That is tyrosine. Awesome. Tyrosine. Yeah. Okay. Is everyone good on that concept? Sometimes polar, sometimes non-polar, context dependent. Yes? Cool. Okay. I'd like you all, in the last 3 minutes that we have here, to think of an amino acid substitution that would almost definitely change the structure of your protein. Like just name. Yeah. And I'll leave up this slide here for you.
That one.
Okay.
That'll help.
I mean, I think the spectrum one might help more. Yeah, that's fine. Yeah. Okay. And talk to your neighbors about it. Tell the person next to you what you're thinking.
We will stay a little later if you have specific questions.
Yes.
So.
Yes. I mean, if it changes.
It is.
We did
actually
not
on
get
the
time
left
for that.
side.
It's on the last slide. If you want to read the slide, it's on the front. We're gonna make an announcement about that.
Oh, well, actually, I looked
Oh,
right at the end.
on the, I think the quiz, it talked about
Oh,
measuring the level of potency. And it was RBP.
it's just like, I was right.
Oh, I got it, whatever. No, but it's like it's actually blurry, whereas, no, not blurry, it's multi, this one is RBP. Wow.
So you like the little
Do you know?
O for the negative sign?
I wouldn't think, I wouldn't strike you as.
You have it?
I think you actually.
I don't know. No, you don't.
I don't think it was for either thing. It was for sure. I don't know why.
But I'll be great.
Oh,
Maybe it was. Wait, what's the argument?
I did, but that was full on.
Yeah, of course.
So you be explaining what's the difference between the polar and the negative charge and then.
So the charge amino acids are technically a type of amino acids, right? But it's like a subcategory because they have polarity.
there's so many
It's
things.
strong polarity, strong enough that it can consider them like having a formal charge. I can talk to you more about that. I'm gonna make an announcement.
It's like 2 weeks old.
That's anything.
Oh,
Exactly.
but, embryonic multiplexes, two lineages, and yes.
Okay, so we're basically out of time. I'm so sorry to tell you that we did not get to stem cells and
Well, you might as well
potency.
just go over it.
Okay, you can read these slides yourself. Potency, right, and the different types of potency. You have totipotent, then pluripotent, then multipotent, then a fully differentiated cell. That's like a hierarchy, not a hierarchy. It's like a one directional, right? It goes in one direction, and it's going down. This is the most potent, and this is the least potent. And basically, any cell with potency is a stem cell. That's basically the important part. That's the definition of a stem cell, a cell with potency. Potency is the ability of a cell to differentiate. And that's it. If you want to stick around and ask questions, you definitely feel free to. And these slides are all posted to Canvas. If you have any questions, please reach out. I'm Amy, that's Ben. And I'm gonna stop the recording now.
Good job, guys.
It's.